All right. Hello, everyone. So for this week, we're going to be discussing metabolism. I've actually combined chapters 22, 23, and 24 into one set of lectures, kind of pick the highlights that are the most important parts of each one of these chapters. And they're going to cover different aspects of metabolism. But that's going to be our goal for this week. Um, for metabolism, similar to a lot of the other biochemistry chapters, there's a lot of stuff that you just have to know, kind of what I would consider almost like trivia type problems. So you're going to want to flashcard it and pay attention also to your mastering chemistry assignments, because I've specifically gone in there and chosen questions that reflect what aspects of this I expect you to know. Okay. All right, so let's get started. We're going to start off with kind of just talking about metabolism in general and specifically about carbohydrate metabolism. Okay, so there's two different types. Metabolism is sort of a large umbrella term that covers all reactions that occur in a cell. And we can break those into two different categories. One would be catabolic reactions, and this is when we break down a large or complex molecule into smaller components, and those reactions provide energy. Okay, so catabolism is breaking down large molecules and providing the cell with energy. Anabolism or anabolic reactions are the exact opposite. These are reactions that require energy, right? But they build larger molecules. So protein synthesis or building glycogen or, even creating new cells. These are all reactions that require energy because you're building something larger from smaller building blocks. Okay, so metabolism is the umbrella term. We can separate that into catabolic and anabolic reactions. Our main focus is going to be on these various catabolic reactions. Those are, how the cell actually gets its energy. That's going to be our major focus. But we will talk a little bit about anabolic reactions as well. Okay, so first of all, when we talk about reactions that create energy, what we're talking about is reactions that in some way, shape, or form generate ATP. ATP is like the currency for energy in a cell. If a cell needs to do something that requires an input of energy, it uses ATP. Okay, so ATP is adenosine triphosphate. Right. So when we talked about nucleotides, we talked about how they had different bases. So this would be an adenine base on a ribose sugar. So the nucleotide is going to be this ATP. If it was being used in RNA, it would be this AMP building block, right? We talked about how RNA is composed of these different building blocks. AMP would be one of those building blocks. If we stick two more phosphate groups on it, right? So 1, 2, 3 phosphate groups, that's how it becomes ATP. Okay. How is ATP used as energy in the cell? Well, because converting ATP to ADP, right, cleaving off one of those phosphate groups is an incredibly favorable reaction. It releases 7.3 kilocalories of energy per mole of ATP, which is a good amount, okay? So again, just going back to our ATP structure here, right, what we would be doing is cleaving or hydrolyzing, really, this phosphodiester bond right here and creating ADP and then that free phosphate group. Hydrolyzing that bond releases a lot of energy. Okay. ADP can also be hydrolyzed and releases the same amount of energy, right? So removing that phosphate group, hydrolysis of that phosphodiester bond is really favorable. Typically, it's ATP that is used in reactions. There are some reactions that use ADP. Typically, ATP is like the go-to energy currency in the cell. Okay. So how does this sort of work? Well, basically, the cell pairs this very favorable reaction, the hydrolysis of ATP, with reactions that are unfavorable energetically, right? So, for example, converting glucose to glucose 6-phosphate requires an input of energy, but pairing it with ATP, the hydrolysis of ATP will release energy. And so it makes that reaction a net favorable reaction, right? So reactions don't occur unless they're energetically favorable, unless they release energy. So if you want to do something complex that's going to require an input of energy, so for example, synthesizing a protein or this simple reaction here, you have to input more, you have to pair it with a reaction that's more favorable, right? So this reaction that the cell uses over and over and over again is this hydrolysis of ATP reaction. Okay, so catabolism of carbohydrates occurs in 3 steps. So first is the digestion step. Step 1, digestion. This is where you break down your larger polysaccharides into smaller monosaccharides, okay? Those smaller units are what can enter into the cell. So the cell can't import a big polysaccharide into it. So the first step that has to happen is within your stomach, and actually it actually starts in your mouth, is to break down those polysaccharides into their individual monosaccharide components. Then they can be moved into the cell. Within the cell, those glucose molecules or those monosaccharides are broken down into 2 and 3 carbon compounds, okay? So they're broken down even further, essentially broken in half in a series of reactions. But yeah, so that's step 2, is to break down those monosaccharides into 2 and 3 carbon compounds. That happens in like the cytosol of the cell, then those are moved into, those smaller components are then moved into the mitochondria, where oxidation of those small molecules takes place and ATP is released, okay? So that first step is kind of the simplest one. This is digestion, right? So when we eat foods, some of the food that we eat already contains sugars in monosaccharide form, but a lot of it contains starches and other sort of polysaccharides. So the first step, and again, it actually starts in the mouth. Your saliva contains enzymes that will break down these polysaccharides, but continues in the stomachs and the small intestines. And essentially all you're doing is breaking up those polysaccharides into their individual components, okay? Those tiny individual components are what enters the bloodstream and then enters into the cell. Okay, so they get absorbed through your intestinal lining and stuff like that, entering into your bloodstream where they get circulated throughout your body and then moved into the different cells as monosaccharides, right? So digestion is all about going from polysaccharides all the way to monosaccharides. And again, that's because those are what can be moved into the cell. Okay, so this is sort of a little bit of a cheat sheet on these different stages here, and we're actually focusing pretty exclusively on carbohydrates at this point. We'll talk a little bit about lipids and we'll talk a little bit about proteins, but our big focus right now is on carbohydrates. So again, those start out as polysaccharides. They get digested into monosaccharides, which can move into the cell, right? So pass through that cell membrane as monosaccharides. And then those monosaccharides are broken up into those smaller components at stage 2 and moved into the mitochondria. So in the mitochondria is when these smaller components are broken down. I'm gonna make a little edit here. We haven't really talked about this acetyl-CoA. We haven't talked about any of these different components. We'll talk about that here, but I do want to update this figure because it's a little misleading. What actually moves into the mitochondria is not acetyl-CoA, but pyruvate. Okay, so we'll talk a little bit about these different components, and that's going to be the entire goal of this chapter, is to pick apart each of these individual reactions. So this figure is probably going to look a little intimidating at first. We'll go back, once we go through these, once you've watched these next, I think 4 lectures, go back to this figure and it'll make a lot more sense. Okay, so just to talk about the cell structure real quick. We haven't really talked about, we mentioned various parts of the cell randomly, but we haven't sort of taken a step back and taken a big picture of the cell. So let's make sure to do that. We know that we have a nucleus in our cell that contains the DNA. So there's not any sort of well, there is metabolism, but there's not any sort of generating of energy that occurs in the nucleus. The nucleus is really just there to protect that DNA. The only thing that occurs in there is the translation of DNA to RNA, as well as replication of the DNA. We have a membrane that's made up of those phospholipids that separates the outside of the cell from the inside. So there's the cell membrane on the outside. And then the part in the middle, most of the cell, the fluid part of the cell, the interior of the cell, is what we refer to as the cytosol, okay? So if we just sort of take a picture here, again, we have this nucleus. This is the cage that houses the DNA, right? Kind of protects it from everything else going on. The cytosol is, again, that intercellular fluid. So what everything's floating around in, the nucleus is floating around in the cytosol, the mitochondria is floating around in the cytosol, et cetera. We talked about ribosomes. Remember what ribosomes do? Ribosomes are what takes that RNA information and actually synthesizes those proteins, those polypeptides based on those RNA codes. Ribosomes are floating around in the cell a little bit, but actually most of them are bound to what's called the endoplasmic reticulum. That's what's pictured here in pink. So you see that if we kind of zoom in, you see that all of our ribosomes are studding on the outside of that endoplasmic reticulum. We're not going to really worry about a lysosome. The cell membrane is, again, made up of phospholipids. We talked about those when we were talking about our lipids chapter. Those are what sort of makes, separates each cell from one another and keeps all that fluid inside the cell. And another big component that we're going to talk about a bunch is the mitochondria, because this is where most of our catabolism takes place, is within the mitochondria itself. You've heard the mitochondria is the powerhouse of the cell, right? Hopefully you've gotten that ingrained in your brain from like high school biology. What that means is that that's where most of these catabolic reactions take place. These reactions that generate ATP take place within the mitochondria. All right, so the different components, so like we sort of just talked about this. So this is just a little cheat sheet of what we just mentioned. All right, so let's do this learning check real quick, make sure that we know the difference between these descriptions for ATP and ADP plus PI. That's PI stands for that phosphate that's been cleaved off of that ATP molecule to create ADP. So which one of these are used in anabolic reactions? Remember, anabolic reactions require an input of energy. So that would be ATP. The energy storage molecule, yes, again, that's ATP. When we talk about creating energy, storing energy, we're storing that in the form of this ATP molecule, right? And it's, again, all about that very favorable cleavage reaction. Combined with energy requiring reactions, this is, again, that's just a fancy way of saying anabolic reactions, right? Energy requiring reactions. So that would, again, be ATP. And the hydrolysis products are ADP and PI. Again, that PI is that phosphate group. Okay. All right, so this is kind of a little bit of a cheat sheet of the different things that we're going to cover. And so I just kind of wanted to give you guys an introduction. We'll take these lecture by lecture for the most part, but just to introduce the terminology, because it's going to be a big chapter here, okay? So glycolysis, this is going to be the first reaction that we learn about. This is once those monosaccharide glucoses make their way in the cell, they're converted first to pyruvate. This step also comes with the release of energy in the form of ATP as well as NADH. And so we talked about ATP. We'll talk about NADH here, especially when we get to the electron transport chain. It's essentially like an intermediate to producing more ATP. So the punchline here is that at this step here, all of the NADH that we create will then be converted to ATP. So the pyruvate that's generated from glycolysis goes through another set of catabolism steps, which converts it from pyruvate to acetyl-CoA. This also generates NADH. That acetyl-CoA will then go through what's called the citric acid cycle, where it will make ATP, that direct energy payoff of ATP, as well as more NADH and FADH, which is similar to NADH. It also will lead to the creation of more ATP, right? So then last in this step is the electron transport chain, where all of that NADH and FADH that's been created is converted into ATP. So again, you can see how all of this will lead to an energy payoff of ATP. And so these steps, these reactions happen back to back to back in order to go from glucose to ATP, okay? The glycolysis happens in the cytosol, in that fluid or within the cell. Everything else happens in the mitochondria. Okay, so that first step of creating glucose to pyruvate, that happens in the cytosol. Then pyruvate's moved into the mitochondria, where the rest of those steps happen. Okay, so these four are all carbohydrate metabolism steps. We're also going to talk about beta oxidation, and this is for lipids. Okay, so this is converting a fatty acid into acetyl-CoA. That can then make its way into this citric acid cycle and then the electron transport chain. So this is when we'll switch gears and talk about lipid metabolism, and that's beta oxidation. Okay, so we'll get acetyl-CoA as well as NADH and FADH. So again, this is now going from a lipid and converting that into energy. And just an FYI, that also happens within the mitochondria.